It's a very nasty disease, progressive deterioration in respiratory function, associated with a continuous cough, and in particular we're looking at what are called exacerbations here every so often, and with increasing frequency as they get sicker, patients deteriorate, produce more material when they cough, and get more breathless. And their sputum is heavily colonized with bacteria. Now, of course, from a medical point of view, all bacteria are bad, so these people must be treated with Agent Orange or something similar to get rid of all the bacteria. And some of the consequences we're going to see shortly. So this is I suppose it's an illustration of the technology that clinicians have to bear, in contrast to the technology that microbiologists have. So these fabulously expensive pieces of equipment that produce these incredible images show you that actually these people can't breathe out. They have narrowed airways, which mean that their air sacs expands and eventually burst. Critically, from our point of view, all the mucus secreting glands in their airways, this is an airway disease rather than a sac disease, produce lots and lots of mucus, which is a lovely home to lots of lovely microbes. And this is just a summary slide to illustrate that at a simpler level. What do the people like, or what do they do to get themselves like that? Well, I think most of the risk factors are illustrated here. Eventually, after you've been in this so-called blue bloater stage, you get to be a pink puffer, which is emaciated because you haven't had enough oxygen to keep yourself going for an extended period of time. Smoking and exposure to smoke is the major risk factor here. There's also a genetic predisposition, which genome-wide association studies are bringing to light. As I said, the exacerbations are widely believed to be infective. And if you look for organisms for which the classical Koch's postulates have been fulfilled in one situation or another, but not in this situation ever, there are no scientific proofs of the individual causality of any individual agent in this situation. What clinicians do is say, yes, we found something that causes pneumonia, so that must be the cause of this patient's problem. If you look at the effects of antibiotics when these people get sick, you basically pay your money and you take your chance. Here's a series of studies which show effectively either a marginal or worse than marginal effect of giving antibiotics, and here are some that show antibiotics are somewhat beneficial. It looks like antibiotics benefit a subpopulation of patients, but we haven't defined that subpopulation, and I guess that's one of the things that we've been trying to do. So I was very fortunate to be involved in a large study described here, in which our respiratory physicians, who fortunately live in the same department that I work in, recruited 156 patients at a fairly advanced stage of this condition, and we observed them for a year and took lots of samples from them, particularly at stable state every 3 months, at the time of exacerbation, and that is before any therapy was started. So these were very well tutored patients to come forward immediately the exacerbation started. We took samples then, started therapy, and then followed them up at 2 weeks and 6 weeks. There was a second phase which I will mention later on. These are the key people. Chris Breitling is a fantastic respiratory physician who set this up. Mona Baffadel is the clinical research associate who did all the hard work, taking samples from patients and taking measurements from them. But here, Karubi Haldar is the microbiology PhD student who did most of the microbiomics. So this just illustrates the sort of numbers that we captured. We did a huge amount of phenotyping at the time of exacerbations. In fact, at most visits, up to 60 features were captured. I'm not going to tell you about all of them. This is the kind of omics that I'm into. This is the sort of volume that patients produce, and you know, it is now many years since I've handled this material myself, but this is the environment that we deal with. One of the issues about this is that it's not a direct sample from where the action is low in the lung. However, the volume of material produced is such that we feel it's a pretty reliable sample, in that when things are coughed up, they get contaminated by the oral microbiota, but the numbers are so obviously excessive in this sort of material that the oral microbiota is completely overwhelmed. Okay. So part of the study addressed these issues in a conventional way, and what you can see here quite nicely is that when you compare people in the stable state and these sort of rogues gallery of well-known pathogens, the difference, if any, between stable and an exacerbation is absolutely marginal, and again possibly a subpopulation. It's widely believed that the total numbers of bacteria increase at exacerbation, and yeah, there is marginal support here, but if you're trying to use an increase to diagnose an exacerbation, it doesn't work the other way around. There's no predictive capacity in this kind of pattern. So the primary analysis was to look at the microbial pathogens by molecular means. We did a total 16S assay, which, when you relate it to the severity of their disease, more severe at this end, less severe at this end, you can see a wonderful relationship emerges here. If you look at the specific pathogens, exactly the same. There's absolutely no relationship showing you that the abundance of a specific pathogen correlates with their symptomatology. So I was going to say we were getting desperate, but we got a nice number of papers out of this and people were faintly interested, so we weren't desperate, but we thought we might like to see whether there were any causal relationships, and I persuaded my colleagues to do microbiomics. In the hope and aspiration that some of these features might emerge, that we might get an undetected pathogen, previously unrecognized. Doing 16S sequencing, clearly that possibility arises. Conventional pathogen that wasn't doing us the favor of growing. Maybe we were underrepresenting or inappropriately representing pathogens. Was there a combination of organisms? There are plenty of polymicrobial infections described where consortia are involved in a specific clinical entity. Are minor components involved, or is a microbiomic shift in itself significant? And of course we're stuck then with whether we're looking at cause or effect. Are the microbes reporting on the environment or shaping the environment? I'm not going to go into this for this community, but we did a standard kind of 16S profiling exercise on a subset initially, because that's all we could afford to do, frankly, at this stage. And these were done by Roche 454 sequencing. We tend to use MiSeq now. So this really, the Findem level shows you what we found, and these are the individual 30 patients through the succession of samples, stable exacerbation, sorry, it's easier up here, stable exacerbation, follow-up and recovery. And I think two things emerge from this. If you look carefully at the individual sequences, there's no consistent pattern, but it's quite clear that individuals are dominated by one or other of these two phyla, the Proteobacteria and the Firmicutes. Looking to see whether there was a profile that particularly mapped to the exacerbation, the answer is there wasn't, and there weren't even any significant changes between the four different states. And on average, there was something like 20 to 30 OTUs identified in every single sample. The rarefaction curves showed that we got close to the limit of what was there at about the 0.1% level. Nothing really very surprising in terms of the range of organisms that were present. Did the diversity change? Well, if you look very carefully at those that were and were not treated with antibiotics, the vast majority were, there is a slight decline in alpha diversity in relation to antibiotic treatments. But in terms of following the pattern of clinical status of patients, it didn't really tell us anything. When you look at the beta diversity and what is driving the community shape, really, 3 organisms that we recognize turn out as dominating the principal component analysis. So these are well recognized agents that are isolated regularly from these patients. Hoping against hope that the more sophisticated the representation we produce, the greater clarity we might achieve. Our hopes were duly dashed. And as you can see, the colours represent the 4 stages. They seem to be more or less randomly distributed across this principal component plot. And the dominant species of firmicutes here and the proteobacteria distributed over a wider spectrum here, but you can see how the individual results map to those genus level designations. At this stage, we wondered whether a description along the lines of that used by Jeffrey Gordon and colleagues in their really extraordinary studies on obesity and other phenotypes associated with microbiomes might be informative. And we looked at the ratio between the 2 dominant phyla, the proteobacteria and the firmicutes. And somewhat to our surprise, a pattern did start to emerge, and we found that we had a firmicute dominated group, a proteobacteria dominated group, and a more or less equal group that came out through a hierarchical clustering process, taking all 4 visits into account. So that could only be achieved on 19 patients because the sequencing wasn't successful on all of our samples. So what we did was we made a qPCR which we calibrated against these results. It correlated very nicely, and applied it to all 30 patients, and the clustering pattern was sustained. So the next interesting question is whether those clusters map to anything that we could measure in the patients. Before we took that on, we asked whether the pattern of what happens in terms of the proteoform relationship might be different in those 3 groups. And what we were immediately struck by was the fact that for the first time we saw a change at the time of exacerbation that was restored during the recovery process in the high proteobacteria group, but not in the other groups. In the dominant, the largest group, the high firmicutes group, the ratio seems to be relatively stable. Interestingly, where the numbers are small, they are in this group as well, something different seems to be happening towards the end, possibly provoked by the therapy. Clinicians like to make generalized linear models to see what is dependent on what, and indeed this was possible with some of the measurements we took, and notably in relation to 2 key indicators of inflammatory status. The relationships driven by the PF cluster fall into a generalized linear model that relates these three factors. And these indicators are considered pretty hard evidence of severity of the inflammatory process going on in patients. If one looks in a little bit more detail at these, you can see the pattern that neutrophils in the sputum samples increase and then decrease in the HP group, but do are less interesting, particularly in the HF group. This C-reactive protein, another important indicator of inflammation, goes up massively here, but far less impressively in this group. So we're beginning to see something that's distinct clinically about this group. So these are small numbers and wouldn't be accepted as anything other than the beginnings of hypothesis generation in clinical circumstances. But in fact, as you'll appreciate, this was part of a much bigger study, so we had a large number of additional samples available to us, and we tried to see whether we could replicate what we'd found in this large group. Well, again, the clustering came out as before with the three groups, and when we plotted changes in the proteoform relationship, again, the high proteo group showed the disturbance and the restoration. So this is, I guess this is something you'd expect. If your therapy was in any way related to what you're trying to measure, it should produce changes that seem to be related to your use of therapy, whereas in this case nothing has happened, and the equivalence group beginning to show this possibly, but with the larger numbers it didn't show this pattern of disturbance at the end that we'd seen previously. So that's looking at the disturbance from a very limited perspective. Can we see some patients in which the beta diversity might also follow a pattern that was reflected in treatment? And the answer is yes, but only in a very small group of patients. So this is part of a subsequent study that we've done where the beta diversity seems to be disturbed and then restored by in relation to treatment, whether it's by the treatment or not, or accidental, I can't honestly say, but one can begin to see these emerging patterns in a subset of patients. There's been considerable involvement in our work by drug companies, and this follow-on study sponsored by GSK, which is still in progress, is really scaling up what we've done. Now, this COPD is a very important disease. It's projected to be the third commonest cause of death in adults worldwide by 2020, given the expansion in exposure to the toxicants that cause it. Given that they're chronically affected individuals, drug companies are very interested because that means you can treat people with drugs for a very long time and get a lot of revenue out of it. And they're prepared to pay for this level of sequencing. So you can see that we're going to get much more detail out of the samples we already have and the ones that we're going to be generating in the future. Again, we're seeing samples dominated by the usual suspects, and indeed we can see the clustering that we noted before in these larger sample sets. We're just beginning to see, with the larger numbers, changes in diversity. This is apparently a significant change, and one of the prominent organisms is beginning to pop out as being related to exacerbations, but there's some very interesting correlations coming from minor components of the microbiota that seem to be correlated with exacerbations. So what I'm saying is, once you push the numbers up, you begin to see more features that are worth investigating. And the final thing I want to show really is that GSK have access to microbiome data from many other sites in the human bodies, and what's quite striking about this is that the assemblages are quite distinctive depending on which part of the body you're sampling. Quite reassuring actually is the distinction of the oral cavity from, these are all our samples, from the lung samples, and that it's not just a subsample of the oral cavity. In our field, really, the idea of bringing ecological theory to bear on explanations of these patterns is really only just beginning, and people are beginning to talk in terms of island biogeography to explain the relationships between these communities. But other than saying it and waving the flag, I can't really add to that at this stage. Some groups have began to describe all the factors that they think are important here, and this is a very nice paper in the Lancet earlier on this year, illustrating some of the points here, that you've got all sorts of ways in which the microbes can be migrating in, ways in which they may be eliminated, and many of these diseases affect these mechanisms, and the hugely complex environmental changes that can go on, to say nothing of the addition of Agent Orange in the form of antibiotics, which clearly have a catastrophic effect. So, application of ecological theory here is really in its infancy. And yes, engineering would be nice, but it would be nice to know what we're trying to engineer. We don't know what is good and what is bad. We've possibly got an idea of what is irrelevant, or we're just at a descriptive stage. And I, certainly be very excited to talk to any and everybody who thinks they may have a theory that is worth applying in these circumstances. I guess we really should be thinking in terms of microbiome management, and one of the thoughts that's been provoked here is, what about the patients in our series who didn't get any exacerbations? Maybe they had microbiomes that were resilient and protected them from getting exacerbations, and we haven't thought about that until today, or maybe yesterday. So that may be something that in the long-term management of these patients we ought to be thinking about, and maybe the introduction of antibiotics is a bit of a catastrophe for some people. So my simplistic conclusions are: we haven't detected a new pathogen, we haven't seen a pattern common to all exacerbations, we've got these three subgroups and the pattern of response to treatment, which is really not a way that anybody has thought about human microbe relationships in the context of clinical practice, at least. And clearly this is a way of stratifying patients, and clearly the next thing we're going to do actually is to see whether exclusively giving antibiotics to the HP group is a legitimate thing to do. So, of course, we've got lots more questions. How can we investigate the microbiome as a causal agent? We could look at what we think might be noxious genes. We could look at gene dosage. We've started to try and use pie crust, which I have to say, although it's very, the prospects are very exciting in terms of just working from 16S data to get some idea of gene dosage. The phylogenetic discrimination is too low to be of any practical use in the data we have at the moment. Clearly metatranscriptomics could be useful, but that's really quite challenging in terms of the sampling. One's clearly got to stabilize the transcriptome within seconds of getting the sample out, and that although we've done it in some areas that I work in, we've not done it in this. We know that microbiomes polarize the immune response, and maybe the balances that we're seeing here will be correlatable with recognizing Th1, Th2, and all the other ways in which the immune response can be subdivided. Are we looking at the causal agent or the effect of the changing environment in these patients? Is the microbiome simply a signal of the environment down in the lung? Other than making that suggestion, I can't comment. But again, it doesn't make much difference if it tells us which patients to treat in different ways. It's still a useful thing to do. And since we have a real-time PCR that could be applied in patient management, that's certainly an interesting prospect. But as with all molecular and microbiome analyses, in practical terms, we need interpretive criteria. It took 50 or 60 years to interpret agar plates in clinical practice, and hopefully we'll be a bit quicker with molecular diagnostics, but it's still a challenge. And just restricting ourselves to the classical Koch's postulates view of infection is clearly inadequate. And with that, I will tell you who our sponsors have been, and thank you for your attention.
Other questions? Oh, yeah, that's the polite. Lady behind Paul, whose name I've forgotten. Yeah.
Okay. I was just wondering to what, if you could comment, and if you know, to what extent the microbiome you recover is similar or dissimilar to CF patients?
Yeah, it is distinct from CF patients. The mucus is of a completely different quality, and in CF patients it's quite restricted in volume and has a very specific composition. We don't see Pseudomonas nearly as much, and they don't show a succession of colonization in the way that CF patients do. And indeed, in CF patients, you can, patients have been around long enough for you to be able to look at evolution in situ. So none of that has really been possible so far in COPD. But we're really only just beginning, and I think the microbiome is more diverse than we see in COPD patients.
Oh, sorry.
Okay, so since you see three groups, that might suggest that it is a polymicrobial infection. And I was wondering, in the medical community, how is this perceived right now? I mean, if we were in the scenario of a polymicrobial infection, I mean, Koch's postulates will not, strictly be applicable, but transfer of agents might be. And so has anybody thought about just basically transferring such, of course you can't do it directly in humans, but to, for example, mice, etc.
Yeah, key aspect of Koch's postulates is you need a suitable animal model, and that's, there are a couple of models which are somewhat debatable, but there would be no reason not to try polymicrobial combinations. In the HP group, they're still very diverse in terms of their beta diversity, so the actual compositions are quite different. It's just the overall ratio between the proteobacteria and the firmicutes is high, so their actual compositions are different. It's just the overall ratio, very, very much in the same way as Jeffrey Gordon's work has shown.
So is this ratio based on relative abundance data
Yes.
that you get, or is it? Have you looked at translating that to quantitative information? That's
to absolute abundance. We've looked at that as well, and it's not as clear when you look at it in absolute. You just multiply by the 16S value you got from those. Yes, so absolute abundance doesn't produce clarity.
So I've got a minute. I was going to permit myself a question. What you actually need to do is to define, before you can define disease, you need to define normality. And traditionally, disease has been the presence of something. It could be, in your case, the absence of something.
Oh yes, sure.
And you have actually exactly the same problem as a meat has, in that you have that. So a meat was using his occupancy abundance curve or frequency abundance curve to define normality. So you might be able to use that approach to define what's normal, and then be able to detect variations from normality, either in your subgroups or in your things as a whole.
Yeah, I mean, that comes back to what I was saying about stability. So maybe the people who don't get exacerbations are the closest that we're going to get to normality. These are inherently abnormal patients because they've got 10 to the 8, 10 to the 9 bacteria per mil of their sputum. And in the normals, as you know from the PhD you examined for me,
5 years ago.
Normal people have far lower communities, which actually map more closely to the oropharyngeal community. But I accept what you're saying, that it's probably more important to define a stable status than it is the unstable status.